Dye-sensitized solar cells, or DSSCs, have been perceived as a solar technology with much potential. Besides being cost-efficient, they have high energy conversion rate and work well under low light conditions. One of the hot areas of research regarding this technology is the quest for a photosensitive dye, a dye that converts light energy to electrical energy, which is both efficient in energy conversion and economic for production. This research aims to explore the use of beta-carotene as a dye in DSSCs and to investigate whether the cells could reach a conversion efficiency of 10%. After a literature review, 3 solvents, ethanol, dichloromethane, and acetone, that potentially suits the need of extraction of beta-carotene was tested in order to find the best one that suits our need, with the highest solubility of beta-carotene and the darkest stain left on the titanium dioxide powder. Dichloromethane solution showed the darkest color of reddish-orange, much darker than the yellow-orange acetone solution and the pale yellow ethanol solution, suggesting that the concentration of dye is the highest in dichloromethane solution. However, the dichloromethane extract resulted in the beta-carotene having staining issues with the titanium dioxide nanopowder. We suspect that this is due to the water immiscibility of dichloromethane, preventing the dye from thorough contact with the titanium dioxide nanopowders, while ethanol and acetone, being water miscible, do not have this issue. Hence, acetone was then chosen to be the solvent of beta-carotene extraction for the relatively high solubility of beta-carotene and the water miscibility. In the research, 2 methods of bonding the dye onto the titanium dioxide layer were used. One method was to soak the conductive glass coated with titanium dioxide layer into the dye solution. The other was to mix the dye solution and titanium dioxide paste inside the mortar. Cells made using each of the 2 methods were tested for their open circuit voltage, and the group of the cells made using the first method gave a mean open circuit voltage of 640 mV, while the other gave a mean open circuit voltage of 598 mV. In our investigation, various ratios of water, ethanoic acid, and titanium dioxide nanopowder were investigated, and the cells were tested for open circuit voltage. The best recipe for the paste was found to be 5 grams of titanium dioxide nanopowder, 10 drops of ethanoic acid, and 10 milliliters of water. In the preparation of titanium dioxide paste, the 5 grams of nanopowder was added first, followed by the ethanoic acid, and lastly water. A thin layer of titanium dioxide paste was then spread onto the conductive side of a piece of conductive glass, and the glass was then heated under a hot argon for 30 minutes. For the extraction of beta-carotene, all the juice in the papaya, which was essentially water and other water-soluble substances, was squeezed out. As beta-carotene is insoluble in water, the juice containing little or no beta-carotene. The beta pulp was then immersed in ethanol to extract other alcohol-soluble substances first. After that, the mixture was filtered, and the residue was soaked in acetone under a warm water bath for approximately 40 degrees for 40 minutes. The mixture was then filtered again, and the filtrate was condensed until saturation. After the dye solution was prepared, the conductive glass with the titanium dioxide layer annealed was then immersed in the solution for 30 minutes before it was taken out, assembled with the anode, which is another piece of conductive glass coated with carbon, and rinsed with ethanol solution. 2 batches of cells, the first made 28 days prior to the test and the other made 1 day prior to the test, were tested for efficiency. Both batches were made by staining the dye on a titanium dioxide layer using the beta-carotene acetone solution. The maximum efficiency achieved was 6.0%, which was significantly lower than 10%. In this investigation, a workable procedure of the making of beta-carotene cells was explored, and optimum composition of titanium dioxide was found. Despite the effort to optimize the cell for the use of beta-carotene, the efficiency achieved was still significantly lower than 10%.